To that end, we're going to move on to our next presenter. So we have Lena Biosciences, who is the CEO of Lena Biosciences. And again, she's going to tell us about another very exciting technology. Go ahead, Yelena, and share your slides.
Thank you, Katie. And you got it there. You were able to say the last name. Okay. So, hello, I'm Yelena Biosciences with Lena Biosciences. We have a commercial technology which replicates human body for systematic drug testing. We are positioning the platform for cancer prognostics to optimize therapies for patients using their own tumor tissue after surgery and ultimately biopsy. We are seeking partnerships with oncologists to demonstrate the value of our lab-ready product in a clinical setting. While the precision medicines market is forecasted to grow to $86 billion by 2025, these drugs can only be used with 5% of patients. And for many drugs, the improvement in survival is measured in months. Why? Precision drugs focus on specific mutation. The problem is that cancer evolves past that mutation, and it evolves differently in each patient. Our Profused organ panel is the solution to identify drugs which work for the patient's own tumor. In our platform, we can grow mini tumors from a patient's surgical resection to identify drugs which kill the patient's tumor en bloc. In our platform, we can save patients 4 to 6 months of time to obtain a personalized drug therapy result at half the cost of growing their tumors in mice. In addition, we can test all drugs, not just those that work in mice, to see what works best for the patient, not the mouse. Here are the features of our market disruptive technology, which can select the best drugs for each individual patient. First, our system grows tumor in 3D. This restores cell shape, receptors, which are drug targets, and cell functions such as invasion and metastasis that only exist in 3D. Second, our system provides perfusion because most tumors bigger than 1 to 2 millimeters are perfused. Third, in-well perfusion concentrates cellular secretome, metabolome, extracellular vesicles such as exosomes, cell-free DNA, and evDNA for therapy monitoring and ultimately prognosis. Fourth, perfusion enables intratumor delivery of monoclonal antibodies and antibody drug conjugates that can only be delivered and tested with systemic blood circulation, which our platform mimics. Fifth, our synthetic hemoglobin blood substitute provides blood-like oxygen delivery for long-term support of immune cells for immune and cell therapy testing. Thanks to the blood substitute, the function of vital cellular enzymes is restored. As a result, in our platform, cellular drug metabolism is increased tenfold compared to cells in a dish to see if the drug really works or it doesn't. In sum, we have a comprehensive test system which can identify drugs that kill the entire tumor, not just the parts of it. And this includes pre-metastatic cells, which eventually make it into the blood. Here's how our system predicts the efficacy of cancer drugs. We have two examples here, a small molecule inhibitor and a large molecule monoclonal antibody, Herceptin. These drugs work in 3D, but not in 2D. When tumors are grown in our system, receptors are restored and drugs work at typical doses we see in clinic. Here's another example. For antibody drug conjugates, our system can answer if linked toxins can selectively target tumor drug metabolism and eradicate cancer, or provide reactive metabolites that inside the cancer cell create overwhelming oxidative stress and kill it. We can answer this because our system increases drug metabolism log-fold compared to cells in a dish. Next, we can answer if targeting cellular antioxidant enzymes can eradicate future metastasis because our system supports pre-metastatic cells that substantially rely on oxygen. Lastly, because the cells in our system are in 3D, we can tell which molecules cause lipid peroxidation or enough membrane damage that leads to endocytosis of efflux transporters that are responsible for drug resistance to primary therapy. Our team has over 60 years of combined experience in tissue engineering and life sciences. I have 3 issued patents and am the principal investigator on Small Business Innovation Grant Awards from the National Institute of Health and the National Science Foundation, which funded our technology developments. Our CSO is an experienced biomedical engineer with prior appointments at Emory. Our strategic partner is Specicare, a tumor tissue bank, which we have been using for the initial validations. With Dr. Dixon, who is the Chief Medical Officer at Specicare, we founded a small personalized therapy pioneer consortium comprising small companies and universities. Our technology developments have been funded by prestigious SBIR awards after passing rigorous scientific review, which speaks to our credibility as a company. We developed our own intellectual property and secured the patents at scarce resources. This speaks to our innovation and resilience. We have patented products and customers in the research market, including cancer research and pharmaceutical companies. Our customers are publishing in peer-reviewed publications using our products. The papers that we publish have thousands of reads. We are a part of the ATDC and JLabs incubators. To summarize, through our innovations, we are driving a new market in personalized medicines. We're looking for strategic investors and partners who share our vision for the future of cancer care. We have a solid track record and are in the fundraising mode. The funds will be used for business development, sales and marketing, 510(k) regulatory device approval, CLIA, and related certifications. We ask for your help to secure partnerships with oncologists and healthcare systems. Lena Biosciences, the future of personalized medicines.
Yelena, this is so exciting. Thank you so much for sharing with us. It seems like this could radically change the way we make decisions about cancer treatment. Talk about how this fits into the clinical workflow.
So our device fits nicely into a routine clinical lab workflow. Any clinical lab has standard cell culture and readout equipment to grow and interrogate cells both quantitatively and qualitatively. And our disposable plates have a standard multi-well plate format, which is the most frequently used consumable in a cell culture lab. The only difference is just the attachment of one tube and the placement of the pump outside the incubator. For chemosensitivity, we use a standard cell titer glow ATP assay, which most technicians use, and we can recommend how to multiplex assays, for example, for fluorescence and luminescence to get additional readouts.
Great. We have many questions in our audience here, so I'm going to go directly to those. 2 of them are about other, I would say, competitors or comparisons. How do you compare your solution with organoid-based drug sensitivity testing? And do you consider your technology a major advance over tissue/organ on a chip type technologies?
So we're actually at the intersection of both. So our device technology, yes. So we are funded by that NCATS initiative for organ on a chip technologies, but we have a few major differentiators. So for organoids on their own, they're not perfused. So we use that technology in a slightly different way. You can still call it an organoid. And in terms of organs on a chip, we just turned that into a high throughput device. So rather than having, just imagine you want to test 96 cultures in a plate, right? So what you need is 96 tubes coming in, 96 tubes coming out. You need 96 pumps or pump channels. It gets really, really complicated. We boiled it down to something really simple when there is just one tube, okay? You still have 96 statistically independent conditions. Just because we have that blood substitute that is really awesome, and it's a special chemical that acts like hemoglobin in terms of delivering oxygen and taking the CO2, yet it's totally inert and doesn't interact with anything in the medium, so we're just basically using it as a little pump. In addition, with organs on a chip, you get a lot of, I would say, a lot of air in the system. It's really, really small, and it clogs. You can't really do it for a long time. We come from that world and had a technology like that. We thought that it was not ready for the market, not throughput scalable, didn't fit into routine, and we figured this is better.
All right, so a couple of questions about this microenvironment that you are creating for these biopsy samples. So clearly maintaining that microenvironment is essential to accuracy of testing. I want you to be able to talk a little bit more again, how you are keeping that physiologic environment where you want it to be. And then a follow-up question to that is, how do you know, in fact, that your blood surrogate is in fact comparable to actual human blood?
Okay, so one thing, so just to say on the side, like how we actually do it and how we are maintaining. So we keep the cells in 3D. So we have a special procedures where we come from tissue engineering. So we can create spheroids, tumoroids, anything. We can isolate the cells, mix and match them. We've done this for years. We take this a step further, in effect, where we try to keep the original heterogeneity of cell types. So the method itself is similar to what Calvin Co has at Stanford, but we have the perfusion, and we're not culturing at the air-liquid interface, because that it doesn't have the physiological exposure. It's super physiological oxygen, right? So we're trying to keep that immune stroma. We try to keep all the cells on block, yet we keep them submerged, but we have the blood substitute. How do we know? Well, we measured actually oxygen consumption rates in the cells. And we do know that even if we had a hepatoma, using the appropriate medium, we can increase the oxygen consumption rate 4 times. So going from cells being completely glycolytic to actually cells utilizing mitochondria. And with that, we were able to go approximately 60% of the lower end of the primary human hepatocyte respiratory metabolism. So we do know that, and we measured, like I said, oxygen, how does it work? And these compounds, these similar compounds have been used actually as blood substitutes. The difference, our difference is that we're not making emulsions. So we don't need huge equipment to supercharge them with oxygen. We just equilibrated, that's covered by the patents. So they always have the exposure, the right exposure to oxygen. Important thing to remember, because the tissue is submerged, right? And it's always perfused in the media. You cannot give it more oxygen than it can take from the environment. And it's the tissue, it's that gradient between the tissue and the oxygen that actually drives it inside. So it's almost like an oxygen hemoglobin dissociation curve. Ours is a little bit more linear, but we have a buffer between the tissue and the blood substitute, which is that medium that makes it just perfect.
And again, the idea being the more lifelike you keep the environment, you're going to be able to get these results. They're going to be more accurate, and you're going to be able to get them much quicker, correct?
Yes, absolutely. So our goal, so if you really create the right environment to keep the tissue, because some of these liquids have also been used in the past for like to maintain organ transplants. So if you really create an environment that the tissue likes, you're actually able to test drugs a lot earlier. And if you miniaturize that, you don't have to wait too long for anything to grow in an animal. You just do it right on spot. So our goal is, with the further validations, to really make a big leap, get a tissue, we don't think that, mutations, if you culture for a short period of time, they're not very likely to happen because they happen more as the cells divide. But what we're really trying to do, this is going to be our next big thing, is to demonstrate that the function has not changed. Then you have a perfect replica. Then you have a tumor in a dish. So, and for that, we will be working with SpeciCare most likely to get the gene expression as it comes out of the patient. When we put it during transport, when we get it in like a day or two in our system, and if we can show that it hasn't changed, boom, that's really huge.
Well, this is super exciting, and I know will be, like I said, transformational for cancer treatment everywhere. So thank you so much for being here.